in the success of ECS. He's made numerous important contributions to our understanding of the structure and function of my favorite ion channel, the IP3 receptor. And so without further ado, I think Jan is going to present an overview of IP3 receptor function regulation. And then Fernanda is going to present some exciting new data relating to regulation of IP3 receptors by metabolic enzymes. So over to you, Jan.
Okay, thank you. So David, thank you for the very kind introduction. It's indeed already a long time I am in the field, and the IP3 receptor is also my favorite channel, as you can imagine. Martin and Adelina, thank you for inviting me, and thank you for the organization of this webinar. The point I will try to make in this short introduction to the work of Fernanda is that the IP3 receptor, of which here you see a 3D structure of the tetrameric IP3 receptor as presented in a recent review from Philip van Betegem, that the IP3 receptor has the properties to do all possible things in the cells, that it's really a calcium channel for all seasons. And before going into that, I want first to acknowledge the team, because obviously all what I want to tell is due to the past members of the laboratory, which are not depicted here, and then the present members of the laboratory who are working together with Geert, myself, and Humbert de Smet on these topics. I will give, as I say, a broad introduction, and then Fernanda, depicted here, will take over and present her recent results. I will also show some results from Nicola here, Ian here, and Jens. And then I want also to thank all the collaborators, outside collaborators, and we have in fact many more, but I couldn't list them all. So these are the more important, I would say, with whom I have worked already for the longest time on this topic. And especially I want to highlight Clark Desselhorst, David Thiel, and Martin Woodman, because the work I am presenting is really indebted to their contributions of all three of them. This being said, I think that for this audience, I do not need to make a large introduction. You all know that so many transporters, channels, pumps are needed to regulate the calcium concentration in the cell. In all the various organelles, there are those transporters, and it is the regulation of those transporters which globally are known as a calcium signaling toolkit, a name which was coined by the late Mike Berridge, and that this regulation allows the calcium signal to vary in frequency, amplitude, kinetics, and localization, and that this is, of course, important to perform the regulation of various processes in the cell. And I will obviously focus on the IP3 receptor. Now, there is one dimension of the IP3 receptor which is really important, I think, is that the IP3 receptors can form signaling hubs in the cell. Because, and that is taken from an excellent review on the IP3 receptor and its regulatory proteins from Colin Taylor, where you see the linearized IP3 receptor with the various domains of the IP3 receptor, and here in the same color, all kinds of associated proteins that can interact with exactly those domains. And if we make the count, we are already over 150 proteins which can be associated with the IP3 receptor. Their binding sites have not always been identified, and it's even more difficult because some of them, calmodulin is therefore an example, BCL2 is another example, have multiple binding sites. Also, we have to realize that multi-protein complexes are possible. It means there can be on the same IP3 receptor various associated proteins at the same time, or at one binding spot, there can be many proteins which makes a complex together on the IP3 receptor. The function of all those proteins needs still to be clarified. It's only for a small amount of them that it's clear what they are doing. And we have to be aware that their function can be dependent on the context, on the cell type, on the differentiation state, and so on. Moreover, it becomes more and more clear that associated proteins can be the target themselves of regulatory mechanism, and so that we have a new level of regulation acting on the IP3 receptor. And for these reasons, I am convinced that the associated proteins greatly contribute to make sure which function the IP3 receptor will perform in a given situation. And I give here just a few examples of various proteins which have been studied to a greater or lesser extent in our group. And these different proteins, as you can see, have all different functions of different roles. Calmodulin can have calcium-dependent and calcium-independent effects on the IP3 receptor. Pyruvate can be released by IP3 from the IP3 receptor and perform a regulatory function on other channels. Polycystin 2 can be activated by the IP3 receptor, and so on and so on. We have proteins performing all different kinds of functions on the IP3 receptor. Moreover, it's very important, and that many of you will be aware of it, that IP3 receptors, which are located on the ER mitochondrial contact sites, will modulate and help the cell make the choice between survival or cell death. Normally, in normal situations, the IP3 receptor will send an appropriate amount of calcium to the mitochondria, and in this way regulate bioenergetics. And this will lead to cell survival. And if not enough calcium is sent to the mitochondria, autophagy will be triggered, which also is a survival mechanism in the first place. But if larger amounts of calcium are transferred to the mitochondria, we have mitochondrial calcium overload leading to cell death. So regulation at this place, the regulation of the IP3 receptor at a given spot in the cell, is really important for the cell. And since this is about the choice between cell survival and cell death, obviously is of importance for cancer cells, and therefore it's not surprising that many tumor suppressors are acting on the IP3 receptor, normally to support IP3 receptor function, while oncogenes, and also a lot of oncogenes, are acting on the IP3 receptors, and they will try to inhibit the IP3 receptor. And in this list, you can recognize BCL2. BCL2 is a member of an anti-apoptotic family of various proteins, some of them being pro-apoptotic, in fact. And this BCL2 has been a topic already for a long time in our lab. The idea to study BCL2 was in fact given to us and in collaboration with Clark Desselhorst, which started for looking into these effects of BCL2 on the IP3 receptor. In short, BCL2 is a relatively small protein containing a hydrophobic groove. This hydrophobic groove binds and inhibits the pro-apoptotic family members of BCL2. BH3 mimetics, because the binding is to the BH3 domains of those proteins, BH3 mimetics will also occupy this groove and so antagonize the binding of those pro-apoptotic family members and lead to apoptosis. And BH3 mimetics, like venetoclax, are used in the clinic to evoke apoptosis of cancer cells. Now, there is also a BH4 domain, and this BH4 domain interacts with the IP3 receptor and inhibits the IP3 receptor, and in that way also inhibits apoptosis. And there if this binding is disrupted and we have a peptide, we can do this, that's BER2, then again we will have apoptosis. Here you see the structure of the IP3 receptor, the three-dimensional structure of the IP3 receptor that is taken from a paper by Irina Seresheva. This is the tetrameric IP3 receptor, of which one of the four units is in color. The different domains are in different colors. And here there is also a model of this monomer where the various domains, the 10 different domains, are color-coded in the same color as here. Now, I will not summarize the work we have done over a decade on trying to understand the binding of BCL2 to the IP3 receptor, otherwise than to show that ultimately what we found is that BCL2 has 3 binding sites on the IP3 receptor. One is an interaction with the transmembrane domains, and we believe this is needed to bring the BCL2 protein in the neighborhood of the IP3 receptor. Then we have a binding site in the middle of the IP3 receptor, in the regulatory region, and it is the binding site of this site that we express as a peptide to compete with BCL2 and so to disrupt the interaction. Dus the BER2 is in fact the amino acids of this binding site. And then recently we found also that there is a binding site in the IP3 binding domain, and there is an interrelation between IP3 and BCL2 because they affect each other. So it's quite a complex situation, and it becomes even more interesting, I think, if instead of looking sideways to the IP3 receptors as we do here, we would look from the top. So from the cytosolic side of the protein, we look down on the IP3 receptor, and then we have this view of the tetrameric structure. And the various sites are color-coded in the same way as on the previous slides. So we have here the interaction, that's the pore domain that we see from above, and we have the interaction of the BCL2 transmembrane domain with the transmembrane domain of the IP3 receptor. Then we have the binding site in the regulatory part of the IP3 receptor. And in green is the IP3 binding domain, with in red the IP3 binding amino acids. And when you look from the top, you see that in fact the binding site in the regulatory domain is quite close to the binding site on the IP3 binding core. And that leads to a number of interesting questions. First question is, what are the dynamics of BCL2 binding? And is there an interplay between the two sites? Do they antagonize each other, or do they support each other? I mean, this is an important question. Another important question is, what about the other proteins which are associated with the IP3 receptor, and of which some of them, like BECLIN1, BI1, CISD2, we know are binding or can bind to BCL2. To which BCL2 are those proteins binding, and when are they binding? The third question is, there exist BCL2 mutants, and it would be interesting to know how those BCL mutants will interact with the IP3 receptor. On the one hand, for knowing what those mutants are doing, but on the other hand, obviously, to better understand what happens here in those binding sites on the IP3 receptor. And finally, another question is, what about BCL-XL binding? BCL-XL is the brother of BCL2, very similar to it, and that is obviously a question to know how BCL-XL compared to BCL2 in the interaction with the IP3 receptor. I will first start with the BCL-XL, and then we will move on with the other topics. Here you see the structure of BCL2, and work from a long time ago, from Giovanni Monaco in our lab, indicated that the lysine 17 in the BH4 domain was very important for the interaction of the BH4 domain of BCL2 with the IP3 receptor. BCL-XL, depicted here, is very similar to BCL2, but on the place where is the lysine 17, we have an aspartate, and consequently, the BH4 domain of BCL-XL is a very poor interactor with the IP3 receptor. Now, in the framework of another study, we found out, together with our collaborators, that there is another lysine here in the BH3 domain, and that this lysine in the BH3 domain can, in fact, take the same role as the lysine in the BH4 domain, because when we make a superposition of those two structures, we see that both lysines, in fact, occupy in space the same place. And this was further investigated, and as I know that Nicolas Rosa, who performed this study, and Geert Bulting, have last year also presented an ECS webinar on the topic. I will just focus on the most interesting experiments, I think. And these experiments of these data have been also published very recently in Cell Death and Differentiation. The first experiment I want to show was performed in collaboration with David Hewlett and was performed in the lab of David Hewlett and shows the open probability of the IP3 receptor in control conditions in the presence of BCL-XL and in the presence of BCL-XL where this lysine 87 is mutated to an aspartate. And you can see how critically this lysine 87 is as the inhibition of the IP3 receptor, which is here clearly seen with wild type BCL-XL, is completely abolished. In the same way, and these are experiments performed in our lab by Nicola, we see here how staurosporin induce calcium signals and induce PARP cleavage, so apoptosis, and we see that BCL-XL is suppressing the calcium effect of staurosporin and suppressing the PARP cleavage, while the mutant, where an aspartate is present, is much less effective. So this shows that lysine 87 in the BH3 domain is important for BCL-XL. And this is summarized here in this slide. That's a figure of a paper that Nicola just submitted, a review paper that he just submitted, and where it's clarified that BCL2 interacts with its BH4 domain and via its transmembrane domain to the IP3 receptor, and that BCL-XL work in a very similar way, interacts in the same domains, but with either due to the lysine in the BH3 domain, or as Kevin Vasquez has demonstrated via an hydrophobic cleft. Then I want to go over to the second point, the mutations, as I thought we think is important for investigating those mutations, and I will just focus on one type of mutations, that are mutations which give to BCL2 resistance to venetoclax. AVR stands for acquired venetoclax resistance. And you can see the structure here of BCL2. You can see the hydrophobic groove with here in orange venetoclax present. And the amino acids lining the groove, so BH3 domain, BH1 domain, when mutated, can decrease tremendously the affinity for venetoclax up to a 200-fold less affinity. It is a project which was taken up recently by one of our bright PhD students, Ian de Ridder, and these are completely unpublished data, so I will just show a number of preliminary experiments, and he expressed various mutations of the BCL2, of those mutations which provide resistance to venetoclax. Calcium traces are shown here. Red trace is controlled trace. Blue trace is in the presence of BCL2 wild type. And the various green colors indicate the effect of various mutants. And as you can see, some are more effective, some are less effective than the wild type BCL2. And now what he tried was to compare those IP3-induced calcium release by those various mutants, to compare them to the effect of those mutants on staurosporin-induced apoptosis. And as you can see, here also we see a variation, and some of those mutants are more protective against apoptosis than BCL2 itself. The control condition without overexpression of a BCL2 is shown here for the calcium response and here for the PARP cleavage. And BCL2, the control wild type, is always in the first row. Another point that I want to show is that the BCL2 IP3 receptor complex forms a hub for various interactions and regulations. So we have BCL2 is a strong regulator of the IP3 receptor, but it can be used or modulated by other proteins. One example is here. It's the IROM1 and IROM2 pseudoproteases, and that has been shown in a very nice paper from the group of Matthew Freeman in Oxford and Colin Taylor in Cambridge about 2 months ago, showing that when this protein is present, BCL2 is losing its inhibitory effect on the IP3 receptor. What I will discuss is not the IROM1 and 2, but another protein which is very interesting for us, called CISD2. CISD2 is a very small protein, 15 kilodalton, containing an iron sulfur cluster, which is redox active, which is known to have various effects in the cell, including on the calcium flux at the ER, and it is localized very interestingly at the ER mitochondrial contact sites. What makes it also interesting is that loss of function mutations are responsible for a rare but fatal disease, Wolfram syndrome 2. And one of the reasons maybe that CISD2 was a little bit less known is that it goes under many other names, including NAF1. There are many more names, but NAF1 is a little bit better known in the calcium community, I think, because of the original work now 12 years ago of the group of Gordon Shore. And in this work, Gordon Shore showed that NAF1, so CISD2, is stabilizing the interaction of BCL2 with beclin, and so inhibiting autophagy. And only when the beclin is released due to another factor, then beclin will move on to provide autophagy in the cell. And by the way, but I will not go deeper into it, we showed about 10 years ago also that this beclin-1 can feed back on the IP3 receptor to induce autophagy. So these are a lot of reasons that makes us that this NAF1, this CISD2, is interesting for us. And another of our bright PhD student, Jens Lonke, is busy to study the effects of CISD2 on the IP3 receptor. He has recently shown that CISD2 and the IP3 receptor can interact, and he has also developed a knockout cell line based on HeLa with the CRISPR-Cas9 technique, where CISD2 is completely absent. And now he is studying how CISD2 affects IP3-induced calcium release, and here it's with a mitochondrial calcium sensor, so we are directly looking to mitochondrial calcium. You can see while ATP stimulates calcium uptake into mitochondria, this is the green trace, in the CISD2 knockout cells, uptake is much lower. So CISD2 works at the level of the ER mitochondrial contact sites and affect calcium transfer by the IP3 receptor. Is BCL2 involved? That is a logical question since BCL2 binds to CISD2. But according to our results, here we have again the wild type cells, here we have the knockout cells, and in both cases, BCL2 inhibits the flux of calcium. So CISD2 has an effect on the IP3 receptor independently of BCL2. These are the results I wanted to show. So what is the take-home message? First, that the IP3 receptor is a target of many proteins that either modulate its function or localization and or are themselves modulated by the IP3 receptor. Now, BCL2, but probably also other proteins, including BCLXL, are central partners of the IP3 receptor. Now, the BCL2 IP3 receptor complex forms a central tenet of several multi-protein complexes, which will regulate IP3-induced calcium release in various contexts. Obviously, it's clear that much more work remains to be performed to uncover the exact physiological role of those various proteins. And very interestingly, we have now exciting work about a new protein, which is completely different of all those proteins I have mentioned until now. And Fernanda, that is the topic of Fernanda in our group. And Fernanda will present her recent results, which I think are really exciting. Meanwhile, I thank you for your attention, and we will take questions, both Fernanda and myself, after Fernanda's talk.
Can you see my slides?
Yep.
Yeah.
I think we're fine.
Okay. So thank you for the opportunity to show my, oh, sorry. Yeah. It's because I can hear the echo.
Yeah, my microphone is closed. I will close the door.
Sorry, guys, it's because I can hear echoes. So today I will present my research in progress. So it's about the metabolic enzyme that we are studying, the PKM2, that can interact with IP3 receptor and can modulate its functions. So as you notice, our group are really interested in finding and characterizing proteins that can interact with IP3 receptor and change their activity. So recently, together with the group of Professor Clark Distelhoff, we published this paper where we used this strategy using this peptide we called D5SD peptide. It's a peptide designed based on the subdomain of the domain 5 of IP3 receptor. It's a region that it's highly conserved in the IP3 receptor type 1 in different species, and also highly conserved among the different IP3 receptor isoforms. We bound this peptide with biotin, and we were able to pull down a list of proteins that could potentially interact and modulate the activity of IP3 receptor. But one specifically protein that really interested us is PKM2, the pyruvate kinase M2. The pyruvate kinase is the enzyme in the end of the glycolysis pathway responsible for the conversion of phosphoenol pyruvate to pyruvate and ATP. We know so far that the gene PKM is able to encode two isoforms of pyruvate kinase, the PKM1 and PKM2. And PKM2, it's highly expressed in embryonic and proliferative cells. And also, recent paper showed that PKM2 it's highly presented in cancer cells. That's why we are really interested in understanding the interaction between these metabolic enzymes and IP3 receptor. So far, we know that PKM2 can interfere in the calcium signaling. So recently, James and collaborators in 2020 showed that PKM2 can form a macrocomplex close enough to the plasmic membrane calcium ATPase in order to provide this pump enough apport of ATP and avoid, in this case, calcium overload and cell death caused by calcium overload. We also know, according to Liu and collaborators in 2017, they showed that PKM2 can modulate the expression of IP3 receptor type 1 and type 3 in different kind of breast cancers. So here you can appreciate that in the absence of PKM2, the cells started expressing the isoform 1 and 3 of IP3 receptor in this kind of breast cancers. We also contributed to this field, showing that PKM2 can interact with IP3 receptor type 3 in different kind of cells, so in B cells, in T cells, and embryonic cells. And moreover, using the PKM2 knockdown cells in OCY LY10 cells, it's a kind of B cells, we showed that PKM2 decreased this calcium signaling here in the control cells. You can show that, I can show you that the calcium signaling is decreased compared to the cells that can not or have the decreased level of PKM2. Okay, so after all this information, we decided to moving on in our project, and right now we are working with this hypothesis where the PKM2 can interact with IP3 receptor in order to decrease the cytosolic calcium signal, and can also modulate the calcium signaling in other compartments, for example, in the mitochondria. So in order to do this, we decided to use the HeLa cells. It's our cancer cells that can express the 3 types of IP3 receptors, and we showed that these 3 isoforms of IP3 can interact with PKM2 here. So using the technique of CRISPR-Cas9, we successfully knocking out PKM2 in these HeLa cells. And we confirmed that our previous results, where we showed that the presence of PKM2 statistically decreased the calcium signaling when you compare the absence of PKM2. So showing that PKM2 definitely has some effect in the IP3 receptor activity. To better understand this difference in the cytosolic calcium, we decided to check the ER calcium content. So we used the Topsy-Gardner, and here you can appreciate that there is no difference between the 2 curves, the curves in the Y type and the PKM2 knockout cells. So it seems that PKM2 is not involved in the control of ER calcium content, at least in our model, in this HeLa cell model. And we also didn't see any difference in the expression, the level of expression of IP3 receptor type 1, 2, and 3 over time. So here we checked different passages of the cells, and no obvious difference were observed in the expression of IP3 receptor. However, in this PKM2 knockout cells, here you can appreciate that there is a upregulation of PKM1. So in a way to compensate the absence of PKM2 and keep the glycolysis functioning in the cells. Okay, so in accordance with our results in the cytosolic calcium signal, we also saw that in the mitochondrial calcium uptake, we we saw that PKM2 can decrease the mitochondrial calcium uptake. So here in the Y type, we have a lower signal compared to the absence of PKM2, here represented in red. And we decided to check if, in addition to the modulatory effect of PKM2, other difference could explain these results here in the mitochondrial calcium uptake. To do this, we started checking the ER mitochondrial tethering in these HeLa cells. So for specifically this experiment, we used the SPLIX sensor. It's a really elegant way to check the MEMS, the proximity between ER and mitochondria. So this is a plasmid where one part of GFP protein is tagged to one compartment, for example, here in the ER, and another part of GFP is tagged to mitochondria. So when these two compartments are close enough, it can change the conformation of the GFP protein in order to promote the fluorescence. And here you can see that in the Y-type cells, or in the cells that still have PKM2, we have more dots. Fluorescence dot means that we have more contact between ER and mitochondria compared to the PKM2 knockout cells. We quantified this, and this difference is statistically significant, and means that it's not the difference that we saw in the mitochondrial calcium uptake between the Y-type and the PKM2 knockout cells is not due to any different, well, we cannot explain due to this any difference in the ER mitochondrial tethering. We moved on and we checked the mitochondrial membrane potential in this HeLa Y-type and PKM2 knockout cells. And for doing this, we used the GC1 sensor. It's when it's in a monomer, when it's in an aggregate, we have a higher fluorescence in the red channel, and it means that the mitochondria membrane potential is higher, so it means that the mitochondria is more polarized, and probably more the mitochondria is more functional compared to the lower aggregates. And here you can appreciate that in the presence of PKM2 in the Y-type cells, we have lower mitochondrial membrane potential. So this difference here can contribute to the difference that I saw in the mitochondrial calcium uptake, because we have this ionic force that can drive the calcium to the mitochondria. But now I want to shift gears a little bit, and we started to generate mutants of PKM2 to better understand the interaction between PKM2 and IP3 receptor. But first, it's important to say that post-translational modification is really important for the function and also for the interaction of PKM2 to other proteins. For example, it's well characterized that the phosphorylation in these specific residues can promote the interaction between PKM2 to beta-catenin, STAT5, and HIF1. We also know that CARM1, it's a methyltransferase. It's an enzyme that it's highly expressed in some kind of cancer cells, can also methylate PKM2 specifically in these residues, in this arginine 445, 447, and 455. And this methylation, it's important for the interaction between PKM2 and IP3 receptor. Here, this result was shown by Liu and collaborators, and what they did, They muted PKM2 in order to avoid this methylation, so they substitute this arginine to lysine, and this non-methylated mutant of PKM2, here you can see that cannot interact anymore with IP2 receptor type 1 and type 3 in this breast cancer cell line. According to the literature, also this the double mutant, the substitution of the arginine at the position 445 and 447, almost completely abolished the methylation of PKM2 and decreased significantly the pyruvate kinase activity. So after showing these results, based on the literature, we decided to start with this mutant, this double mutant of PKM2. So we substitute the arginine at the position 445 and 447 to lysine. And we successfully transfected HeLa cells. In this case, it was HeLa PKM2 knockout cells. Just to make sure that to mention that the plasmid that we used was the PKM2 tagged to GFP. So here, that's why you are seeing green staining for the both white type and the non-methylated mutant of PKM2. We also co-transfected with a marker for ER, and we saw a kind of co-localization that is expected, and it's already described in the literature. But for now, we didn't see a huge difference in the distribution and co-localization of PKM2 compared the wild type and the mutant version. However, when we check the function of this PKM2 mutant, so we had a really interesting result. So we used the HeLa PKM2 knockout cell again, and we reconstitute the PKM2 white with the PKM2 wild type or the PKM2 non-methylated version. So here in blue and in green, respectively. So here you can appreciate that comparing to the empty vector, the PKM2 wild types, the cells that were recomplemented with the PKM2 wild type showed a decrease of mitochondrial calcium uptake. On the other hand, when we reconstituted with the non-methylated version of PKM2, we could not see this inhibitory effect on IP3 receptor. And another surprise that we have is that this mutant can still interact with the isoform 1 and isoform 3 of IP2 receptor. So here we checked using the immunoprecipitation technique. We checked the interaction, and we didn't see any difference comparing the cells that were transfected with the wild type or the non-methylated version of PKM2. So just to summarize this part of the result, we saw that the non-methylated mutant of PKM2 cannot interfere in the IP3 receptor activity, but can still interact with IP3 receptor. So it brings more complexity to this interaction. Okay, so just to partially conclude, because it's a research in progress, I showed you today that PKM2 inhibits IP3 receptor activity and also decrease the mitochondrial calcium uptake. PKM2 can affect the ER mitochondrial membrane tethering and decrease the mitochondrial membrane potential. And this non-methylated mutant version of PKM2 cannot inhibit IP3 receptor activity, but can still interact with IP3 receptor. So I would like to thank all my lab mates, but especially Professor Jan Parys, my supervisor, and Professor Butnik. And my collaborators, Professor Martin Butman, Professor Clark Distenhoff, and Professor Gianluca Bartella, and FWO for the financial support. I'll be more than happy to take questions.
I'd like to thank Jan and Fernanda for great talks. So questions, anybody? Kevin.
Yeah, so in the second talk, the last part about the apparent depolarization in the mitochondrial membrane potential, how do you think that is, what's causing that?
Well, it's a good question. It's something that it's described in other kind of models of the absence of PKM, or it's something that it's already described as an effect of PKM2. Maybe it's something related with the metabolism. Yeah, it's something that I can think. Yeah, I need to check this better, but yeah, some difference in the mitochondrial metabolism can explain this difference. It's something that I have in top of my head.
Sandip?
Hi. Yeah, that was really nice. I just wondered, you mentioned in the introduction that this enzyme may be fueling the plasma membrane calcium ATPase, right? So could the potentiated signal that you see when you knock out this protein be secondary to that, i.e. you've reduced the clearance, and that's why the calcium signal is bigger in the cytosol, and secondary to that, that's why there's more calcium uptake into the mitochondria?
And I forgot to mention, but the way that I analyzed the calcium and the cytosolic calcium, it was in the absence of extracellular calcium. That's why I think it's a direct effect on IP3 receptor. But in the physiological condition, yeah, definitely.
But even in the absence of extracellular calcium, the pumps will still be working, right? So I wondered if you looked at the rate of clearance and see if in your knockout cells, so once you get the calcium spike, is it more sort of sustained, or does it come down at the same rate as it does in the control cells? That might address whether the pumps are affected.
Yeah, it's a good question. It's something that I need to check individually in the cells, because the way that I presented the results, I used the average of all the cells, but maybe it will be clear if I analyzed cluster of the cells. Yeah.
Yeah, great. Thank you.
Sorry. Yeah, thanks.
Professor Marlin?
Okay, I have actually two questions there. The first one is whether the methylated mutant is actually still active in converting phosphoenol pyruvate to pyruvate generating ATP, and does it affect the mitochondrial the mitochondrial ER junction?
According to the literature, this double mutant has decreased of pyruvate kinase, but we don't check in our hands, because in our case, we saw in this PKM2 knockout cells, we saw an upregulation of PKM1. So we are not sure that it's something that I will check, definitely, the pyruvate kinase activity after the transfection with this non-methylated mutant, because I don't believe that we'll have a huge difference in the the pyruvate kinase, because we have upregulation of PKM1, that it's more effective to produce, it's more active to produce ATP. About the ER mitochondrial compound, it's another interesting thing to check, because we just check in the absence of PKM2. We just compare the absence and the presence of PKM2, but not after the reconstitution with this mutant. It's a really good question, and definitely I will check this. Thanks.
Thank you.
Cecilia?
Thank you. Yes, I have a question to both speakers. Is it known whether a change in the cellular redox state will affect the interaction of these proteins with the IP3 receptors?
That's a very interesting question, Cecilia. It is known that the redox state will affect the IP3 receptor activity, but it's one of the gray zones on the association with those proteins. Little is known, and certainly redox state is certainly not something that many people are checking, because it's rather difficult to do. But that is certainly for some proteins like CSD2, it would be interesting to check that.
Yeah, for the PKM2, we know that PKM2 has a function of reducing the ROS, the stress, oxidative stress in the cells. So it's another way that PKM2 can contribute to cell survival, to cancer cell survival. And yeah, it will be really good to check. After reconstitution with this non-methylated version of PKM2 or other mutant that I'm working right now. It's a really good point. Thanks.
Thank you.
So I may have missed this, but PKM2 interacts with all three IP3 receptors. Whereabout does it interact?
That's the million-dollar questions. We would like to know it. We have results which are very interesting, and which were in fact obtained by Clark Disselhorst in the first place. Now, this Clark was studying the interaction of the LCK kinase with the IP3 receptor. And it is by using the peptide which disrupts LCK interaction with the IP3 receptor that eventually he found out that PKM2 was another target. Mm-hmm. And LCK is highly expressed in T cells, but for example, in HeLa cells or in HEX cells, it's absolutely not expressed. Also in most B cells, it's not expressed. So the idea now is that the same site which some cells use for binding LCKs with the IP3 receptor, in other cells, PKM2 would bind to the IP3 receptor. But as we have seen that many proteins have more than one binding site, we are also considering that other target sites are present and that PKM2 is maybe binding simultaneously to two sites or separately to two sites. And we are doing experiments on that, but these are complex experiments. We need pure proteins for doing that and pure fragments of the IP3 receptor. And then we will use BLI and MST techniques to sort it out. So probably a site similar to LCK, and that is then, how would I explain it, quite in the cytosolic part, but quite near already to the transmembrane domains, but potentially also another site which would be, according to some preliminary results, also in the neighborhood of the BCL2 binding site, and making it even more interesting for us, because PKM2 has also been shown that it can phosphorylate BCL2, and so we are thinking about a model where the PKM2 binding to the IP3 receptor would affect the BCL2 binding to the IP3 receptor. But at this moment, this is pure speculation.
I'm sort of interested in your idea that there's so many things binding to IP3 receptors, and just because they do bind might not mean that they do anything in a particular cell if there's something that binds to a similar site with a higher affinity. So,
yeah, that's right. But also it can be that it binds, but that another protein which should at the same moment bind is not present, is not expressed, and therefore the effect would be nil. So we have to always take into account the full interactome for each of those proteins, and obviously that's a lot of work. So we are working now at proteins where we see some relation between them, so that we can use data from one project to support a next project, and therefore BCL2, Backlin-1, Bax inhibitor 1 are all part of the same project. But now we have the impression that PKM2 can relate also to this interaction of BCL.
Great. Sandip, you've got your hand up again.
Yeah, just related to that point, Jan, about potentially shared sites. So, you said with BCL2, you kind of hinted that the sites on the IP3 receptor are in close proximity, right? And so maybe it simultaneously binds to both sites. Have you considered a docking approach? Looked at the dimensions of these various interactors? And of course, there's AlphaFold now with structures if you haven't got the structures, although BCL2 is very well characterized. Would that give you some idea to help you with your more physical experiments?
Probably yes, and no, we haven't done it yet. And there is something we have also to take into account is that PKM2 can exist in a monomeric, a dimeric, and a tetrameric state. So we have to take that also into account. And the effect of methylation onto conformation and so. So there are a number of variables we have to keep in mind, but it's something at some point we have to do, yes.
Thank you.
Any other questions or comments? Well, if not, I'd like to thank Fernanda and Jan again for a really interesting, stimulating conversation.
It was a pleasure.
It's good.
Thank you for the opportunity.
Thank you.